children. So in this video today I will be discussing about two concepts which are slightly different from complete dominance, which was seen by Mendel as a result of his monohybrid cross. The two variations of Mendel's monohybrid cross leading to complete dominance. One is your incomplete dominance and another one is your codominance. So these are the two concepts that we are going to discuss today. Now regarding incomplete dominance, let's take the example of snapdragon flower. You can Google and see the image of snapdragon flower. So snapdragon flower, they come in two colors actually. One is red color flower and another one is your white color flower. So it is known that red is dominant and white is recessive. So in order to represent red color flower, we will write the factors as capital R, and in case of white color flower, small r and small r. So this is your P1 generation. From here, when P1 gametes will be formed, 50% of the factors will be sent over. So from here we have capital R coming in, from here we have small r coming in. When both the factors will meet, this is the genotype of F1 generation. Now, if we go by Mendel's law of dominance, this plant should have given us red color flowers. But in case of snapdragon flower, it is seen that instead of red, in the F1 generation, we find that all the flowers are pink in color. That is, an intermediate character is formed between red and white. Now if we do selfing, as discussed in my earlier video, it is crossing with another plant of the same generation. So from here two gametes will be formed, two factors capital R and small r. From here also two factors will be formed, capital R and small r. So these are the F1 gametes. And if you look at the cross, this capital R, capital R, small r, capital R, small r, and small r and small r. So this will give us red color flower, this will give us pink color flower, this will give us pink color flower, and this will give us white color flower. So in case of incomplete dominance, what do we find? We find that in the F2 generation, there are three phenotypes. We have red, we have pink, and we have white. All three phenotypes coming out in ratio 1:2:1. This is known as the phenotypic ratio. Regarding the genotype, we have capital R, small r, and small r and small r. That too in ratio 1:2:1. So this is the genotypic ratio. So what we find, the basic difference between complete dominance and incomplete dominance, in case of complete dominance, the phenotypic ratio in the F2 generation was 3:1 and genotypic ratio was 1:2:1. However, here both phenotypic and genotypic ratio are 1:2:1. Now we need to understand why this happens. Because now that we know the concept of gene and how they control the character, gene expression, you will be getting in the next chapter, gene expression, how proteins are formed by the information stored in the genes by the process of transcription and translation. So every gene controls a particular protein. And in case of dominance, what happened? The recessive gene is actually suppressed by the dominant gene. So only the dominant protein is expressed. But here basically what happens, none are dominant over each other. So the dominant gene will also form a protein and the recessive gene will also form a protein, and the character which will be expressed will be an intermediate between the dominant and the recessive trait, and therefore we are getting this kind of a ratio, that is your incomplete dominance. Next we will be discussing about codominance. So in case of codominance, we can take the example of human blood group. Now human blood group is determined by a gene represented by I. Immunoglobulin, it's a protein, and this gene has three forms, or we can say three alleles are there. So there are IA, IB and IO, which can also be written as small I. Now the dominance pattern between these three alleles are IA and IB are equally dominant, and both are completely dominant over IO. Now if we look at the different combinations which can be possible out of these, suppose we have I, so this we can write as the genotype, and what will be the subsequent blood group or the phenotype. If the combination is IA homozygous in nature, blood group will be A. If it is IA and IO, blood group will be A, since A is dominant over O. If it is IB and IB, it is B blood group. If it is IB and IO, it is also B blood group because B is dominant over O. If it is IO and IO, homozygous recessive, then the blood group will be O. Now in case if it is IA and IB, then the blood group will be A and B, AB blood group. That means what? Unlike incomplete dominance, in incomplete dominance we found that it was an intermediate character, but here both the characters are equally expressed. That means both the genes will fully express to their full potential. Full proteins will be formed, and therefore, so this is the concept of codominance. Now if we take an example of a cross, suppose we have a question that show a cross between two parents having heterozygous A and B blood group, or it can be also asked that predict the blood group of the next generation. In that case also we can write this way. It is mentioned heterozygous. So heterozygous, suppose one parent is heterozygous A, so the parent will have IA, IO blood grouping or the genotype. Another parent heterozygous B, so IB, IO. Now from these parents, when factors will be formed or gametes will be formed, there will be two combinations possible. It can be IA or IO gamete. From this parent also two combinations will be possible, IB and IO. So what we are having here, IA, IB, IB, IO, IA, IO, and IO and IO. So what we find here, this offspring will show AB blood group, this person will show B blood group, this person will show A blood group, and this person will show O blood group. So in the next generation, there are 25% chances for occurrence of every blood group. 25% of the offsprings will be AB, 25% will be B, 25% will be A, and 25% will be O. So the probability is of 25% that every 4 blood group may come in the next generation. So that is how we are going to solve such questions. So that is the basic difference between codominance and incomplete dominance. So in my next video, we'll be discussing about linkage and how linkage is opposite of independent assortment. Thank you.